Hello everyone, and welcome to today's webinar, Simplify the Development and Implementation of Diagnostic Testing in European Laboratories. My name is Elena Zlatini, and I'm a marketing specialist in Genetic Testing Solution Division for Thermo Fisher Scientific in EMEA region. Today's educational webinar is presented by Laboratories and brought to you by Thermo Fisher Scientific. We encourage you to participate by submitting any questions you may have for our speakers during the presentation. To do so, simply type your question into the Ask a Question box and click Send. We will have a live Q&A session at the end of the presentation where we will try to answer as many of the questions as time allows. You may also submit any technical issues in the chat box if you have trouble seeing or hearing the presentation. Now, I would like to hand over the time to Aspinall from Health Catalysts Group, who will provide an introduction of the speakers and the presentation. Mara, over to you.
Thank you so much. It is great to be here today. As you just said, we are here to answer your questions, but we're going to talk about simplifying the development of diagnostic testing in a clinical lab. Before I begin, I want to put out a disclaimer here for Thermo Fisher Scientific. We appreciate their sponsorship of this event, but as you can see, they're not endorsing, recommending, or promoting any use or application of Thermo Fisher Scientific products. So I'll give you a moment to read that. And before we start our event, what I'd like to do is ask for a poll, and all of you should see this on your screen. We'd like to understand the relative level of experience in this group. So my question is, in the last 5 years, how many molecular tests have your lab developed? And even if you are not the person who is developing the specific test, but you're in a lab that has developed tests, can you please fill this out? If you're not a lab developer, that you can skip the question. 3 options. You've developed no tests, 1 or 2 tests, or 3 or more tests. And I'll give everybody just about 30 seconds to answer this question. And then we will all view the result, but it will give us a sense of how much experience in the group, so we can customize our presentation today as much as possible. Just 10 more seconds. Thank you very much. And when we see our group here, what you could see is we have a number of people, just over a third, who have not done any molecular test development, some with 1 or 2, and then just under a third with 3 or more. So thank you for that, and we look forward to our presentation. So when we talk to our presentation, we have 4 learning objectives today. One is we will go through the basics of describing the different types of in vitro diagnostic tests. Most importantly, we will talk about the difference between an IHD, an in-house, and an IVD, in vitro tests. We will list the benefits and some of the challenges of running either an IVD or an IHD, and we will explain at a high level how these tests are created and regulated. So this will be a good basic understanding, especially for those who have not developed a test or just developed a few tests. For those developing more, we will actually go into some details that we believe will be important. Our core, as we've talked about, are 7 questions. We will go through that diagnostic test terminology to ensure we all have the same baseline. And then we will ask these questions briefly. What is an IHD? What is an IVD? Why use one versus another? Who typically uses one versus another? When and how to develop those tests? How do they compare? And lastly, and critically importantly, how can a lab protect the important work they do and their intellectual property? So as we do that, I'm very pleased to introduce our speakers. I didn't give my background, but I'll very briefly say that I'm Managing Director of Health Catalysts Group, Professor of Practice at Arizona University. My whole career has has been based in the business of clinical and research diagnostics. I had the privilege of serving as president of Genzyme Genetics and CEO of Roche Tissue Diagnostics. But today, we have two speakers who are true experts in the diagnostics community worldwide. Dr. Marilyn Owens and Michael Donovan, Dr. Michael Donovan. Marilyn is a senior healthcare executive with more than 40 years of extensive management experience, establishing and leading clinical reference labs, including academic medical centers, private and hospital labs around the world. She has broad scientific background in immunology, infectious disease, oncology, genetics, and molecular biology. She has substantial lab operating experience with P&L responsibility in large and small labs. Her experience, as I said, spans the world. So she has run labs in the US, in Europe, and in Asia. She has done startups, M&A, and strategic planning. She's an experienced teacher, mentor, coach, and graduate training in business as well as the science. Michael is equally impressive. He's a registered patent attorney specializing in patent prosecution and intellectual property licensing and commercialization with a specific focus on science-related technologies. He has an academic background in microbiology and a PhD in immunology. After leaving private practice, he worked as intellectual property counsel at the Translational Genomics Institute, also called TGen, and recently served as system director for intellectual property at Common Spirit Health. Today, he's a lecturer and head of program at ASU's Biomedical Diagnostics program, where he teaches in a variety of areas, including regulatory law, health economics, and bioeconomics. So now over to you.
Thank you, Mara. This is Marilyn Owens, and I'm going to start off with some real basic definitions. We all know that laboratory tests are considered in vitro, which would be in the laboratory in a test tube. But the in vivo tests are also extremely important because these are driven by physicians. Physicians do EKGs, blood pressure. They will do the imaging studies. And all of this is extremely important for diagnostics. For the in vitro side, we have choices of IVDs or IHDs. And you can see that they are pretty much the same type of test. They are used to detect or quantify levels of desired biomarkers of a various number of types. But the difference between them is that the IVDs are driven by products. These are prepared by vendors and manufactured to certain standards. Whereas the IHDs, in-house developed test, are driven by the process at that particular laboratory. Now, the differences between those two are that the IVDR is regulated by CE IVDs, and these require a certain amount of hoops to jump through in order to sell products to clinical labs for the use in consumer and patient care. They have standardized equipment qualifications and procedures and training. These are regulated by the laboratory industry as well as the government. And these tests must be pre-validated with a specific type of data analysis and bioinformatics report in order to be qualified and clinically validated before they're approved. With the IHDs, these are developed by individual laboratories, and because of that, they can only be performed in the laboratory that designed it and validated it. They can't be transferred, licensed, or sold, with few exceptions. The instrument qualification and training requirements are established by those individual laboratories. So the laboratory sets up its own test and says when it's working and when it's not. But it should follow ISO 15189. At least starting in 2024, that will be a requirement. Right now, it's a recommendation. They're often developed in-house by necessity because there is no CE-IVD test available for what they need. And these IHDs must be clinically verified by running them on known patient diagnoses and can be implemented quickly for emergency use. The molecular tests can include a wide variety of assays for different applications. And those of you who have developed IVDs probably have gone into most of these markets. I think oncology is a major one. Of course, pathogens, genes, and rare disease proteins. Pharmacogenomic markers are becoming very, very hot. And then you also have the respiratory pathogens like COVID, for example, various genetic variants and metabolites as well. But those are just examples. Reasons a laboratory might choose to purchase an IVD test is essentially if it does what you need it to do for your patient population. It has all of the manufacturing done. The controls are in place. There's a broader distribution. It's easier to establish as a gold standard because you can compare it to other labs using the same product. Design controls. Importantly, it has notified body guidance and regulatory expertise. So it's set up to meet the standards of your environment. It has adverse event reporting as a requirement and a complete clinical validation prior to marketing. The reasons a laboratory would choose to develop an IHD, is that the IHD is unique and there's no CE-IVD assay available for sale for the same clinical need. And it might be a clinical need in a different range of detection, for example. That's one reason that you might modify an IVD and create your own IHD. You can't scale up an IVD for a desired throughput, and so you need to modify. IHD would be an improvement over an existing assay. It doesn't align with a desired procedure or utility that your clinicians require. It can be developed much more rapidly than an IVD test. And sometimes this because of pandemics, for example, and the need to create assays more quickly, an IHD is the preferred choice. It can also help a laboratory remain competitive. Now, advantages of IVDs, again, are that the quality system is in place. And so you don't have to essentially set your own parameters. Oh, whoops. I don't know why you can't see me. I'm sorry. I can't control that. Let me talk again about advantages of IVDs. The inventory control is simple because you order a kit, everything you need comes, and you can maintain your own inventory. The technical support is available, so the supplier can troubleshoot and replace any sort of faulty product. The clinical validity of the test has been proven, and therefore it can ensure that it detects or measures the analyte that you're reporting and that is being used for your patient care. There's a broad distribution of IVDs because of the marketing and supply chain of the vendors. So many laboratories utilizing the test can compare their data and can increase or potentially reduce confidence in it. So it's kind of like having your own support group. Advantages of IHDs are that you do have control over the content because you've designed it for your application. And health institutions can select specific and relevant targets and applications for their patient population. You can adapt it, or you can implement it rapidly, modified relatively quickly to respond to market needs. Now remember, any modification, you still have to document and prove, but you don't have to go through as much detail as an IVD did. You can consolidate IHDs into one test. Testing for multiple analytes with multiplexing makes it a much faster diagnosis and can be very helpful for syndromic panels. A laboratory qualification is that your health institution, first of all, has to comply with the general safety and performance requirements, including IHDs. But with IVDs, the quality management system requirements are not mandatory until 2024. However, ISO 15189 is recommended. Now, in kind of a broad view, this is the typical IHD process from planning to launch. And you start with setting up your lab and accrediting it. And this would be whatever your national or institutional requirements are. It can be different in different facilities because of corporate management rules and quality systems. It can be different in different countries. But in general, you have to follow the IVDR Annex 1. Why run an IHD? Well, if you go through the planning process, you say, what do I need? Is it available? And can I make it? What is the test or panel? You would configure that test depending on what test menu you're trying to complement or put together. You would select the targets that are clinically useful for your patients. You can customize it depending on what sort of equipment you have. You would interpret the results in the manner that, and report them in the manner that is most helpful to your clinicians and benefit, obviously, patient care. For the analytical validation, you will do a classic validation following probably the CLSI guidelines, which would cover how you would determine an adequate sensitivity, specificity, reproducibility, accuracy, and also prove that you don't have interference from known components that might be in the same patient sample. And you would split samples for clinical verification. If the tests are clinically accurate, you've verified it clinically with samples from patients with known diagnoses or samples that have been previously characterized blindly by another laboratory running a validated test. Results would be compared to check the concordance. This is also one of the reasons running proficiency tests with exchanges between laboratories. Best practices for IHD introduction for a test launch. Well, you need to let people know that you're working on it. So you have prepared your market. You can announce the test when it's ready. Describe the utility in educational content, forums, publications. And in this peer-reviewed publication, if that's consistent with your IP strategy, which Michael's going to talk about next, I think that laying the groundwork for the test launch actually is ongoing throughout the entire analytical and clinical validation process. Michael?
All right. Thank you, Marilyn. All right. Good afternoon. Good morning, everyone. As Marilyn mentioned, I am Michael Donovan. I am going to, there we go. I am going to talk about two different sets of legal issues that are very important for diagnostics: regulation and intellectual property. First, we're going to talk about regulation. We're going to continue that dichotomy that Marilyn with the contrast between IVDs and IHDs. And again, remember, IVDs are more product driven, and they're about regulating a thing, in this case, a diagnostic, whereas IHDs are more about the process in terms of what is going on in a clinical laboratory. Now, all of you probably recognize that as of May 26th this year, IVDR went live, and devices, or in vitro diagnostics in our case, that are marketed to third parties are now subject to a whole slew of new regulatory hurdles. Some of the big issues, some of the new regulatory architecture to which IVDs are subjected to are harmonized standards from standard setting organizations, including ISO 13485, which is a quality management system that is in place for medical device manufacturers. There is also, depending on the classification of a given IVD, there's significant compliance with conformity elements, as well as appropriate regulatory approval. Now, significantly, one of the big shifts from the IVDD, which is kind of the precursor regulatory system, to the IVDR is the implementation of a risk-based classification. Previously, there were simple categories, and if your IVD did not fit into one, you were basically left to self-regulate. Now, with these risk-based classification systems, which I'm going to talk about next, you have a whole new set of regulatory obligations that are associated with this process. And then one last thing just to mention on IVDs is some of the post-marketing surveillance and controls, adverse event reporting, as Marilyn mentioned. Now, in contrast to that, IHDs, on the other hand, are not explicitly defined under IVDR, but they are also not excluded. Rather, they are regulated by implication under Article 5.5 of the IVDR. Those of you who do roll out IHDs are regulated by a competent authority in your particular jurisdiction. Now, one thing to keep in mind is IHDs under the IVDR are only permissible to be developed and commercialized by health institutions. And that is a term specifically defined in IVDR as an organization whose primary purpose is the care or treatment or the protection of public health. So these are things like hospitals, clinical labs, public health laboratories. These are organizations that are there to protect the public health. So those are the only organizations that can actually roll out an IHD. IVDR, additionally, in 2028, so this is forward-looking at this point, will eventually require justification for the use of an IHD, and I'll talk a little bit more about that in a bit. But in terms of the process that is regulated, again, there's performance documentation that's required. There's the same ISO standard that Marilyn mentioned, 15189, which is quality management for clinical laboratories, as well as that particular laboratory's institutional, regional, and national requirements. Now, as I mentioned, one of the big changes is the implementation of risk-based classification present in IVDR. I'm not going to go into too much detail here, but this risk-based classification is based on a combination of risk factors tied to both public health risk as well as personal risk. And that risk is closely tied in with what an incorrect or an erroneous result might give rise to. So Class D is actually the highest risk category, and Class D covers certain things that like tests for transmissible agents in the blood supply, as well as biological materials that might be transplanted or re-administered. Also, blood grouping and tissue typing, such as ABO systems, rhesus systems, KID, and Duffy testing. So those are the highest risk. And then you slowly move down in terms of lower risk technologies, such as those that have high personal risk. So again, this might be something where you have, you're looking at infectious disease diagnostics or cancer diagnostics or cancer screening, as well as some B, which is a little bit, again, less risk, where you're looking at things like cholesterol testing, urinalysis, as well as certain exempt self-tests like pregnancy and fertility tests. One important aspect to note in terms of Class B is that that is the default category if for some reason your test doesn't really qualify under any other categories. And then finally, we've got Class A, which is basically, broadly speaking, laboratory devices. For example, PCR machines would be considered non-sterile Class A. Also things like wash buffers, other instruments, and specimen receptacles. So again, the really important take-home point here is that you have these different risk classifications, but to go along with that, you also have different levels of regulatory scrutiny that are placed by notified bodies. So the regulatory scrutiny for a Class D device is going to be much more significant than for a Class A device. And in fact, some of those Class A non-sterile devices are actually default to self-certification, kind of the old way of certifying under IVDD. Now, just briefly, let's see here while this pulls out. So the class selection process, how do we figure out into which class an IVD might be classified, is actually initially, the burden is initially on the manufacturer. So it is up to the manufacturer to follow a predetermined set of rules in order to figure out which class their device falls into. And these rules basically ask certain questions about what the device is intended to actually test for. So right here in Rule 1, as I mentioned, some of the highest risk associated devices are going to be things like blood screening, in particular for infectious diseases that could be easily transmissible through the blood supply. So those are very high risk. That is going to be a Class D. And again, you're going to have the highest level of regulatory scrutiny in terms of what those notified bodies are actually going to do. And then you proceed down from basically left to right in terms of asking these questions and delivering various answers until you get down to Rule 6, where basically you have a kind of none of the above type of answer, in which case you default to Class B. So again, there's some great information out there in terms of these specific questions that have to be asked, but you do need to ask these questions in order to figure that out. And then once you make that determination and submit an application to a notified body, they will verify your analysis as a manufacturer. So one last note on IHDs in Article 5.5 is there are actually 5 specific requirements that fall under Article 5.5. The first one, Marilyn touched on, which is you're required to comply with Annex 1, which is the General Safety and Performance Requirements, or GSPR. That actually makes up, I think there's 23 or 24 GSPRs, and within those, there's actually subsets, so there are lots of requirements to keep in mind there. In addition, and Marilyn also touched on this, devices cannot be transferred to another entity. So basically, a health institution can develop and roll out an IHD, but on the other hand, it has to stay within that health institution. And to kind of dovetail with that, you have these IHDs, again, are processes, and they cannot be scaled up to a large industrial scale. They have to remain on that local level. Again, though, IVDR is new, and it'll be interesting to see how industrial scale ends up being defined from a regulatory process. And then in addition to that, you have quality management systems requirements. ISO 15189, which, as Marilyn mentioned, is not quite required yet. It will be in a couple years, but it is something that is highly advisable to follow so that you have good compliance procedures in place. And then finally, justification is going to be required. This is not the case yet. It kicks in, I believe, 2028. But for health institutions that want to roll out an IHD as of 2028, if there is a CE-IVD already on the market, that health institution is going to have to put forth a justification for the development and commercialization of that IHD. So it is something to keep in mind. As of right now, that's not necessary, but it will be down the road. So next, I'm just going to touch very briefly on some intellectual property issues. There are basically two sides to IP as far as from a simplistic standpoint. There's obtaining coverage on one hand, and then there's going to be strategy and potentially avoiding other people's patents on the other hand. With respect to obtaining IP protection, the European Patent Office is kind of what I'm going to focus on in terms of their rules and regulations. It can be quite challenging depending on what your assay is and what platform you're using for your assay. If you're using conventional technologies and you're using conventional or previously known biomarkers. So for example, if you're going to develop a qPCR, a new qPCR assay for BRCA1, BRCA2, it's going to be a challenge to obtain patent coverage for that because those are obviously, qPCR is well known, as are those two markers. Or an inventive step tied in there, but at the same time, you need to be able to demonstrate that to the PTO, or sorry, to the EPO. You may be able to obtain protection for novel platforms. So, for example, if you are able to come up with some type of new way of performing these diagnostics, that could be protectable, and/or improvements to conventional platforms. Those things can be protected. But the big issue in the European Union is the statutory prohibition on protecting diagnostic methods. There were actually, it is one of it is explicitly recited in European Patent Office regulations that you cannot protect a diagnostic method that is performed on a human or animal body. And there were a couple of key decisions coming out of the EPO's Board of Patent Appeals that actually set forth in terms of figuring out what is a diagnostic method. And it might sound common sense at this point, but as of 2005, it was not necessarily so. In 2005, it was set forth that basically a diagnostic method is a collection of data, it's comparison of data, it's an analysis of the data, and then it is a diagnosis based on that data. So those are the key steps that have to be present for that preclusion to actually apply. And then in 2008, there was actually a bit of a reprieve, in particular for the in vitro diagnostics industry, where you have, again, the same judicial board setting forth what does performed on mean from a human or animal body perspective. And this gets back to actually Marilyn's first slide, which was something that is performed in the laboratory. So as long as the diagnostic methodology is performed on a specimen from a human or an animal body, as opposed to actually being performed on a human or animal, it is something that doesn't trigger that prohibition. So putting someone into an MRI tube, that's going to be something that will trigger the prohibition. Taking a specimen like a biopsy and running maybe some genetic screening for melanoma, that's something that will not be prohibited so long as you don't explicitly recite requirements for actually contacting that human or animal body. Now, on the flip side, we have pre-commercialization concerns in terms of freedom to operate. This boils down to avoiding patent infringement, which can be quite expensive for your organization. As such, as early as possible in the development process, it is always a good idea to engage counsel, either internal counsel or external counsel, to conduct basically a patent search to figure out what patents are out there in the particular technological space that you are looking to exploit. The results of that search may lead to either the preparation of certain legal opinions, such as invalidity or non-infringement opinions, or it may even end up requiring your organization to in-license patents if the owners of those patents are willing to make a deal to help you avoid infringement concerns in the future. And one final note is this FTO process can actually provide some good guidance in terms of obtaining patents. You might be able to find something out there that may not pose an infringement concern, but could still inform us in terms of what protection you can and cannot get. Now, with that, I will turn it back over to Mara, as she is going to touch.
Thank you, Michael, and thank you, Marilyn, for a fantastic overview of the key issues around the lab. I'm going to end our presentation piece, leave time for questions to talk about a topic that maybe you've had enough of, but I'm going to give a very brief overview of COVID-19 diagnostics and how that is interacting with our world. As we look at COVID-19 diagnostics, I'm going to show what has changed over time. It started that the diagnostic was a very invasive one, a bronchial lavage. We moved to PCR and viral antigen tests for active disease diagnosis. We then moved to screening, and I believe in the future it will all be about surveillance testing as the active stage of the pandemic moves forward. And what I mean by that are air quality monitoring, breath tests, COVID sniffing dogs, scratch and sniff tests, and wastewater. Most importantly here, wastewater. And what's been important about those is that they moved from, as I mentioned, very invasive tests in the lungs to the back of the nose, to the front of the nose, and ultimately most of these screenings will be passive and non-invasive with saliva or even breath. Where are these tests happening? From the hospital to the central lab, to the point of care, to home. But I want to emphasize here, because most of you are in central labs. While I believe that most of the testing will go home in terms of volume, there will always be an important place in the central lab for PCR and other molecular tests for COVID and other infectious diseases. But for COVID in particular, as it moves to the lab, out of the lab, for the majority of the tests, not the most aggressive ones, we will see the price go down. So how many tests are out there? If you look at this, and this is from testing.com, which is maintained by Arizona State University in the US, and I've worked on this, you can get a sense of the number of tests, just over 450 in the US with EUAs, but the CE European self-certification that you're all familiar with has approved the most number of EUAs or authorized those. A comment here that there are a number of those that are antibody tests, which is the vast majority. In the US, while we authorized a lot of antibody tests, many of them were then pulled back from authorization. So to give you a sense here is the vast majority of our tests are indeed molecular from labs like yours, COVID tests, but we see that 88% are PCR, then isothermal, we have 2% sequencing and 1% CRISPR. I believe that PCR will continue to dominate that field, but we'll have a small number of tests in the lab that are full sequencing based or CRISPR based. How does COVID compare? And while this isn't about diagnostics, I felt that this was a very interesting perspective to look at how COVID has changed over time. This is a look of the worst and most challenging epidemic threats that our world has seen going back several hundred years, and looking at case fatality rate and infectiousness or transmissibility with an R naught. And you can see how we started at a higher fatality rate and moved to a less and less fatality rate in the US and much of Europe, equivalent to the flu. However, Omicron is much more contagious. This is critical, obviously, for society, but I'm going to say it's also critical for testing, because testing, especially the very accurate PCR testing, is the only way to slow a surge. What we know about COVID is that very many people have COVID, but do not have any symptoms, but are still transmissible. And testing is the only way to find those people. So to summarize and give our conclusion, IHDs are a very important instrument for diagnostic innovation. The IHDs leverage regulations that provide labs the ability to quickly adapt to changing needs, and that's so important. The IHDs, as Marilyn emphasized, function through lab processes, while IVDs are licensed as tests, and labs really need to recognize and optimize the processes and procedures to do this well. IHDs and IVDs can work together for patient care, and as we heard from Marilyn and Michael, continue those tests to support medical research and development. And both types of tests make personalized medicine and better care available as a reality for patients. So with that, if you have any questions, we're going to talk about them live, but I wanted to share all of our email addresses. And if you have questions to follow up afterwards, please feel free to email us. We will now go to our live Q&A and see what questions. Please submit your questions in the Ask a Question box. And with that, I will give you a moment to look at that. We've had a couple of questions come through initially. Michael and Marilyn, I'm having a little bit of trouble with my system. Can you call up the first question?
Sure. And sorry for the technical difficulties in my presentation. I did not realize that my video was freezing quite as much as it was. But
we heard you fine, Michael, so it worked out.
Okay, that's good. So the
At least they can see you.
The first question is: laboratories in the European region are still allowed to develop in-house assays under the new IVDR. Can you confirm? So I can answer that, which is yes, they are. The Article 5.5 of IVDR makes clear that there are permissible opportunities for health institutions to develop IHDs, but at the same time, everything you do still have to comply with those rules in Article 5.5, and more importantly, not everyone can develop them. You have to meet the definition of a health institution.
Thank you for that.
Sure.
Next question that came in was about the presentation and existing documents to recap what we went through and any additional details. Michael, I think you've done some work on that.
Yes, there is. I mean, I can actually refer you to the Thermo Fisher team as well, because we have our group put together some good publications, some white papers on these various topics that we've put forward.
Super. The next question, Marilyn, goes to you. Do I need to justify the development of an IHD now or under the new IVDR if there already is a CE-IVD assay with identical specifications on the market?
I would say no, just based on the definition of an IHD. Michael, what do you think? If there's some modification, perhaps, but if it's exactly the same, I don't think you can develop an IHD.
So for the short time, I think it's until 2028 you're allowed to, and then 2028 is kind of where the IVDR comes down on IHDs. That's where you really kind of see the limitation there, where you need that justification before you start developing, because it is. I think that there is, just from my personal perspective, I think there is this bias against IHDs in the IVDR. It's a lot of letters. That there is more and more roadblocks being put in the way for IHD rollout.
I think a lot of those roadblocks actually came from large medical institutions, which didn't want to take on any legal questions. So that was the basis of my answer.
Sure. No, absolutely.
Super. So let me ask a question here on the difference for labs who work in the US and in Europe. How do they manage that? Are there any differences in terms of, probably to you, Marilyn, in terms of setup for labs that they're based in Europe, but they'd also like to have their test available for a US audience?
Yes, that's generally a corporate decision, and it has to do with the quality system that the company is using. But in general, large organizations standardize and would like to do the same in all of their facilities to provide the same patient care. If it's a question of a one-off, I think you could do different methods, but it sure wouldn't be as easy to control.
Fair enough. In terms of that laboratory system, putting that system into place, what are one of the, what I'll call always and nevers? Because we know about 40% of our audience have not developed a test in the past. Is it critical to have that laboratory system in place when you go through the initial part of your process? And if you're upgrading your system, what are some good watch outs that you should think about in terms of putting in a new system?
When you say system, are you talking about LIS or workflow process?
Yes. Yes. I was actually thinking about the LIS system.
Okay. Well, you can use multiple LIS systems if you have labs in different locations and they are managed by different LISs, as long as they can capture the relevant information. And you can standardize your quality control. You can standardize the percentage of positivity you see in a certain patient population. And you can actually compare with proficiency testing between facilities. So the LIS really is a vehicle to document results and patient information. I don't think it has to be standardized for a company.
And how long does it typically take to put in a new system, and how much training is required?
Wow, that is a very, very difficult question. If you have a small laboratory and one technology, to implement an LIS for a single test can take you between 6 months and a year. And if you have a large laboratory with multiple technologies and many different tests, you're looking at 1 to 2 years.
Got it. But I'm assuming that most of the larger laboratories probably have some sort of LAS system in place. So it's probably more about training new employees and upgrades.
It's training users. It's if you're putting new tests into an LIS, there's a lot of software testing that has to go along with that to document that the workflow is capturing all of the right data and that the results that go through various platforms of testing come out at the end with a quality controlled result going to the clinician and or the patient.
Super. So I'm going to switch a little bit. Michael, a question to you about legal status. You've talked about the intellectual property. I often get questions about trademarks and trademarking test names. For many labs, they really want to establish that their lab is doing something unique or putting together a unique process with service to the customers. How does the trademark system work in terms of creating a unique name for your product?
I mean, the trademark system is it's much more forgiving than is the patent system from a inventive step novelty perspective. I mean, a trademark is in general viewed as kind of consumer protection, a consumer protection based policy, so that you have a mark. It could be a trademark, which is generally associated with goods, or a service mark, which might be associated with some of those IHDs like we talked about. But basically, as long as you are the one who is the first to use that mark, or someone else may have ceased using that mark and you have kind of picked up the torch, if you will, it is a good way to try to establish a brand, especially in those situations where you might not be able to get patent coverage. So if you've got a great brand, obviously still complying with all the regulatory aspects that I talked about, but you launch a test, even though there might not be patent protection available for it, trademark protection is still something that can set you apart from competitors.
Super. Thank you. And now we'll go back to the regulation. We have a question from Italy. For those products that will not be, quote, certified IVDR, but it was a CE-IVD, for instance, some probes for fish analysis, how will the lab have to manage that situation? Again, not be certified IVDR, but was CE-IVD.
So depending on the class that they fall into, so something like that, I would think it would be class B. There will have to be basically recertifications under IVDR. So things that are already on the market that had previously been regulated under IVDD, or I guess kind of not regulated under IVDD, kind of fell through the self-regulation loophole that was present there. There's and it's tied into the risk categories. There is a gradual phasing in of those having to go through that regulatory process. So Class B, I think, doesn't have to be phased in until, I think it's 2026 or 2027. So eventually those will have to be to comply with IVDR, just not quite yet.
Super. Marilyn, anything to add there?
No, I have to just say that, for fish analysis, you're looking at some pretty straightforward reagents. And so I think what the laboratory can do to protect itself is to do extensive analytical validations on those probes to be sure that the sensitivity and specificity is correct. And then, Michael, I think it's just a few little steps to go further.
Yeah, no, I mean, the regulatory burden for, I think, like I said, I think this would be a Class B for the probes themselves. I don't think the regulatory burden is all that much. I mean, especially if they've been in use and you've got plenty of data already on the use of those probes. I don't think it would be that much of a hurdle. I think that there's going to be just more quality management as well as potentially post-marketing surveillance that you're going to have to comply with.
Super. So with that, I'm just going to put back up the screen with email addresses. If you have any questions, please be in touch. And Elena, over to you to conclude. But thank you, everybody, for being with us today.
Thank you, Mara, Marilyn, and Michael, for your time today and your really informative presentation. We'd also like to thank Laboratories for hosting today's webinar in their platform. And of course, thank you all for joining us today. Please don't forget this webinar can be viewed on demand. Laboratories will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who might have missed today's live webinar. Thank you and goodbye.